Boa tarde, bem-vindos a mais um webinário da Academia Brasileira de Ciências. Essa é a 36ª edição de nossos webinários. Certamente vai ser muito interessante. É uma homenagem muito especial ao Ivan Izquierdo, que faleceu recentemente, e o título é O Legado de Ivan Izquierdo para a Neurociência da Memória. Um notável pesquisador brasileiro, foi meu colega na diretoria da Academia Brasileira de Ciências e guardo dele memórias incríveis, já que o tema dele é memória. Mas ficou...
Wonderful memories of him, and we miss him very much because not only because he was a great scientist, but as a director of our academy, he was the voice of wisdom, and he had a very strong conscience on the importance of science for the development of our country, and he was able to help all the politics, and with his wisdom, with his knowledge and his perception of the needs of this country. So we miss him a lot today, and this webinar celebrates his contribution and the great person he was. And the amplitude of his contribution is shown by the participation in this webinar, because this is an international webinar, and we will have our Dr. Beatriz Passani, a teacher of the University of Firenze in Italy, and Professor Diana Jarolinski from Buenos Aires, and Carlos Alexandre Neto, professor of the Federal University of the Rio Grande do Sul. This webinar will be coordinated by Esper Cavalheiro, who was the first student from Ivan Izquierdo. He was his very first student. So certainly it's going to be a very important webinar, fascinating theme, which is the memory, the theme of memory. And Izquierdo contributed a lot. He wrote several books. He wrote one fantastic book. He wrote a fantastic book of scientific release, giving an idea of the advances of the science in this field. I would also like to tell you about our next webinar. The next one will be taking place next Tuesday, and webinar that will be the first affiliate section from 2020. These affiliates are the young researchers that come to our academy, and before they are 40 years old, they stay with us for 5 years, then they go out and they come back after they're 40 years yours for another 5 years. So we'll have several presentations from these new members of our academy. This will be a way to compensate this typical thing that is going to happen, what's happening now during pandemia, when we cannot have presential meetings. Our first section was going to be next Tuesday, May 18, and our topic will be environment, new energies, and ecology, which webinar will have the participation of 3 or 4 affiliated members. The first webinar of the series is very relevant for Brazil, not only for Brazil, but for the world, and environment, ecology, and energies. So I would like to see you all there, and to see all the new ideas these young people have. And the other Tuesday, we will have we'll have the number 37 edition of our seminar on May 25th, and we will talk about algorithms, artificial intelligence, and our main topic will be algorithm in charge of our society. This is going to be coordinated by our academic Virgilio Almeida, a member of our academy, and as participants we'll have Ricardo Mendonça, Jana Matthews, and Mr. Filgueiras. So I would like to invite you for this webinar, a fundamental theme, very important for Brazil today, that has attracted international attention. So with no further ado, I would like to ask Mr. Esper Cavalheiro to resume our session for today. We will have 3 speeches, and then we will have a Q&A section. Esper, you have the floor. Thank you very much, Luiz, our president. I'm very happy to be here today. It is an honor, a big satisfaction to coordinate the seminar. I was not really his first student. I was the first student here in Brazil. He already had students in Argentina and other places. But for me, it was a very unique situation. I didn't know him before when I met him, but this quickly became a very important situation because of this wonderful, charismatic person, intelligent, had an absurd, prodigious memory. I think that's why he went to work and he focused on memory because of self-comprehension. And I learned really a lot with him. And I believe that everything that he taught me, I applied. And at his time when he was quite young, and he had a view of science, not only from us, from South American, but the European, from the world. I can only have as a reference, as an image of a leader, of a mentor, as a friend that for me was very, very important. That when I met him here in Brazil, at medical school, when I was looking for some mentorship, and this was the best thing that could ever have happened to me. So in this afternoon, we are going to dedicate to the legacy of Izquierdo. And on behalf of the Academy, I would like to thank our 3 speakers. As Dr. Luiz mentioned, we'll have Dr. Beatrice from Italy, Dr. Diana from Buenos Aires, and Carlos Alexandre from Porto Alegre, that represents in a certain way the places where Dr. Izquierdo worked, disseminated his idea, and he conquered a legion of fans and people that were attached to him, not only for the science, but also to his friendship that was so easy to attain. Carlos was one of his students. I don't know the first or the second one in Rio Grande do Sul. Diana worked for him for decades in a collaboration between Argentina and Brazil, and they sustained this partnership and friendship, and where his family also worked as scientists in Argentina. And Beatrice as a representative, international representative of the great collaborators from Italy. But he had a special passion for Italy, and this maybe took him and maybe led him to call and to work with Beatrice, where they produced splendid works. So today we're going to pay tribute to Dr. Ivan Izquierdo, and I hope that he feels, and you know, that we are paying tribute and then homage to him. I would like now to ask Beatrice to start. Eu queria agora pedir.
In Italy. We thank you very much, and you can give your talk. Do not forget that we have 15 minutes to each one of you. And thank you very much. I'll leave the podium for you, if I can say that using the internet.
Yes, thank you so much. I hope you can see my presentation, hear my voice. It's a great honor to celebrate my dear friend Ivan Izquierdo, to whom I really owe a lot of inspiration in life and science. And I like to remember one of his sentences: somos exactamente lo que nos lembramos y también somos aquí lo que no queremos lembrar. We are exactly what we remember, and we are also what we do not want to remember. And he once said that that he became fascinated by the concept of memory after reading the short story Funès de Memórias by Borges. And
I'm sorry, Professor. I'm afraid that you haven't shared your presentation.
Yes, I did. It disappeared. I'm really sorry. Can you see it now?
Yeah.
Yes. I'm really sorry about that. Sorry. Yeah, it was on the different screen. Sorry, I will start again. And just to express my gratitude for having me here, and in memory of my dear friend Ivan, to whom I really owe a lot in life and science. And I would like to remember one of his sentences: somos exactamente lo que nos lembramos y también somos aquello que no queremos lembrar. So Ivan Izquierdo, he once told me that he gave an interview saying that he became fascinated by the concept of memory after reading the short story Funès de Memórias by Borges. And just by reading from the book, Borges says that this guy had learned English, French, Portuguese, and Latin. I suspect, however, that he was not very capable of thought. To think is to forget differences, generalize, make abstraction. And in the teeming world of Funès, there was only details, almost immediate in their presence. So this kind of memory is absolutely useless. And complete and perfect memory is not impossible. And so, memory refers to the ability of doing several things: encoding, storing, recalling, and extinguishing information that is not necessary, especially extinguish bad memories that can interfere with our life. So all this gives us the ability to adapt from previous experiences as well as to build relationship. And so this is exactly what Ivan dedicated most of his research. And he had the first intuition back in 1986 that the histaminergic system is important, is an extraordinarily powerful, has an extraordinarily powerful effect on memory processes. And he used animal models to understand the process of remembering aversive memories, using these animals that learn to avoid a compartment of their cage because they remember that there they received a punishment. Same thing here to the right on the this other kind of type of model, when the animal learns not to step down a platform because that's a threatening and dangerous place. But at that time, Ivan Izquierdo and Almeida had they thought that histamine was not necessarily a physiological neurotransmitters. Just briefly, the histaminergic system sits in the back of our brain in the hypothalamus. There are a cluster of few neurons that send projections to the whole brain. And together with him, he was the pioneer. He wrote many, many papers about the importance of histamine during the acquisition, during the consolidation of fear memory. And so we began to be interested in histamine as well. And we joined forces. And so we wrote a few papers in the last 10 years. And just using pharmacological manipulation of the histaminergic central system, and we found that during the several learning paradigms, manipulating the histaminergic system, we can modify the animal's behavior and modify their memories in a persistent way. So the take-home message of our story is that the specific action of histamine, of brain histamine, depends on the timing, the location, the localization of histaminergic receptors, and also what brain region becomes activated by the histaminergic projection, and the nature of the cognitive task involved. And he also was the pioneer in finding the intracellular pathways that lead to a permanent modification of neurons, of histaminergic neurons. So now we know that the role of brain histamine is multifarious. The histaminergic neurons that sit here in the hypothalamus project to important region for memory, the hippocampus, the nucleus basalis magnocellularis, the amygdala, the cortex. But these pathways are activated at different times during memory acquisition, consolidation, and extinction. But I would like to start very quickly where the Italo-Brazilian connection started. And I have to thank our dear friend Fernando Benetti, who came to Italy as a graduate student from Ivan's laboratory. We wrote the first paper showing the procognitive, promnesic effect of compounds that are now on the market, H3 receptor antagonist for the treatment of narcolepsy. And these compounds, we found they improve memory in the aversive memory in animal models. And then the Brazilian-Italian connection based mostly on culinary events started. Patrizio Blandina, my husband and mentor here in Italy, he got together with Ivan a grant from the Ciências Sem Fronteiras. And every year, or even twice a year, we visited Brazil, making sure that every time we were there, we had a party going on, celebration of all kinds. And you can see here, Ivan had a bunch of beautiful, cheerful, excellent young scientists that now are around the world doing brilliant science. So the first paper we wrote together, Fernando Benetti was the first author and Patrizio the last author, showed that if you ablate pharmacologically histamine from the brain of these animals do not remember their task. They do not remember that they have been exposed to a dangerous environment. But if you replace histamine in these depleted animals in single nuclei, like the amygdala or the hippocampus, you reinstate the memory of these animals. And this happens in also a time-dependent fashion. So these two nuclei, the amygdala and the hippocampus, take over the task once one of the two nuclei are somehow compromised. So we came to understand with that the histaminergic neurotransmission is critical to ensure memorization of emotionally salient events through the recruitment of alternative histaminergic circuits. So here come my dearest beloved friends whom I miss so much, Cristiane Furini, who's one, and they were both in Florence, and Josiane de Carvalho Mischief, my dearest friends. We wrote together another paper. We looked at the effect of antihistamine that we usually take as anti-allergic to relieve the symptoms of allergy, rhinitis. And in this paper that was published not long ago, the first author was Roberta Fabri, a graduate student in our laboratory. They also joined Ivan's and Cristiane's and Josiane's laboratory. Well, they demonstrated that pyrilamine, which is one of this gold standard anti-H1 antagonists infused in the hippocampus, it impairs retrieval of emotional memory. So it is known that these compounds that pass the blood-brain barrier can affect your memory. So if I can just just give an indication of what the translational clinical value of this first part of our research is, that an inefficient histaminergic system that cannot elaborate peripheral or internal signals may contribute to the onset or progression of neuronal diseases, or to the lack of efficacy of therapeutic interventions. Ivan, with his beloved wife Ivana, came to Florence for the European Histamine Research Society. And then here comes into play Gustavo Provenzi, who is now the pillar of the laboratory. And they began to concoct a new set of experiments on extinction of bad memories. We want to extinguish the bad memory because they can be maladaptive. They can create maladaptive responses. Think about the post-traumatic stress disorders of soldiers in combat, women that have been raped, or other traumatic event that may devastate your life. So Gustavo and Ivan, Patrizio, and all the others came up with the idea of testing a simple kind of medicines, the inhibitors of carbonic anhydrase, which is a ubiquitous metalloenzyme, usually used clinically as diuretic, acetazolamide, anti-glaucoma, anticonvulsant, etc. And so here, Sheila, my dearest friend Sheila, who was a graduate student in Ivan's laboratory, she came initially to Florence for 6 months, and then she stayed for 2 years. So we still hope that she comes back. And what they found is that the inhibitors of acetazolamide, so the carbonic anhydrases, are involved in memory. And their study indicates that the activation of carbonic anhydrase potentiate extinction, so helps you forget bad memories. So this could be, again, a new therapeutic target for development of molecules with less side effects than the acetazolamide or brinzolamides, but the target could be developed subsequently. So just to conclude, the last few slides, food for thoughts. We always had good food for having good science, but also we have good thoughts about food. So it's a story, our story with Brazil, from the table to science and back. And here is Gustavo at Bento Gonçalves with Fernando and Patrizio drinking wonderful wine. The last set of experiments, we dedicated our time to understand if a healthy diet is good also for memory. So what we call the Mediterranean diet, that is based on olive oil, fish, avocado, nuts, etc., is full of omega-3 polyunsaturated fatty acid, which have been suggested to have a potential clinical value in the prevention and treatment of psychopathologies. We tested this in adolescent rats in a model of early life stress that increases the risk of later affective pathologies, including depression and anxiety. So I don't have to go all over the experimental setting, but this is a work that was done with Alessia Costa and Barbara Rani. They are 100% Italians, but they have been around doing their research in other laboratories very successfully. Diana and Gustavo were the principal investigators. So these animals were subject to a stressful protocol, and I will not give you the details, and then they were divided in animals that were stressed with fed a controlled diet or an enriched diet. These were adolescent animals, and they grew up to show their cognitive impairments, like in the contextual fear conditioning. As you can see here, take the bar as a measure of memory. The stressed controlled diet rats were impaired in their memory, and this memory impairment lasted until adulthood, but the diet prevented successfly this memory impairment. Also,
2 minutes.
Yes, I'm included, so I can skip this. And I would like to conclude by showing the last, this is the legacy of Ivan. So we found that the promnesic effect of the diet depends on the integrity of the brain histaminergic system. And we are trying to figure out what's the message between the guts and the brain that allows this improvement. Just to finish, I would say I think this is really the Italo-Brazilian legacy of Ivan Izquierdo. Many young, fantastic, motivated, bright young people that are populating the laboratories all over the world. And here's Sheila, Alessia, Barbara, Gustavo, Cristiane, Josiane, and all the people whom I've met in the laboratory of Ivan. And I would like just to celebrate. I think he would have loved to be celebrated this way.
Thank you, Beatriz. It was very beautiful. Thank you very much for
Beatriz,
your talk.
thank you so much for your... Beatriz, muito obrigada.
Jerusalinski from Buenos Aires, who will talk us about another aspect of the working on memory, the science. Diana, are you there? Can I see you?
Yes, I'm here.
Okay, you can start. I will inform you 2 minutes before the ending. It's okay for you?
Okay.
Okay, see you.
I will share my screen now. Can you see it?
Yes, we can see it.
Okay. Well, thank you very much for the invitation. I'm so glad to be here to honor and homage my master, Professor Ivan Izquierdo, and also a close and dear friend. I would like to say that one great lesson from Professor Ivan Izquierdo was that with merely a few well-selected behavioral tasks and a wise use of pharmacological tools with a deep knowledge of pharmacology as he had, and the precise timing and placement for the surgical approach, many fundamental pathways and mechanisms underlying memory formation, mainly consolidation of memory, have been revealed. Well, this is just to show you the phases of memory, and beginning with acquisition. Well, of course, now is more this than this, the acquisition of knowledge, and is safer to be like this. And then also the skills. And after acquisition, which is the phase of learning, new experience, if there is consolidation, we could have long-term memory storage. And this long-term memory could be evocated sometime. And then this, when evocated, becomes to label stage that could be reconsolidated, which is sort of new learning. I'm sorry. And the star actually in our system, as you know in general from the work of Ivan, is the albino rat. And we use this as a model, and we train the rats. And after some time, minutes and for short-term memory and hours, about a day or more, we perform a test session, and then you can assess the memory as a long-term memory. And we use mainly two tasks in general with Ivan. The open field, which is an open box where the animal could move, freely move and freely explore. There are squares in the floor, and you can follow with a program, for instance, tracking of the travel trajectory, and see where the animals spend most of the time, the speed, and compare the resting time, freezing, and so on. And in this, you can also quantify the rearings, which are the vertical exploration of the space, and the groomings, which is the stereotyped behavior of the animal. And if you quantify this, you can see that in the test session, which is the first bar, crossings and rearings decrease in the test session. In the training session, they are higher, significantly higher than in the test session after some time. It means that the animal recognizes the environment and habituated to them, to that, and respond less with less exploratory behavior. Then you can study there the spontaneous exploratory behavior, the stress in level in an indirect way, the locomotion, spatial memory, habituation to a novel environment. The other task we use with Ivan for long years was the inhibitory avoidant task in one step, which consists in a box where Beatrice showed this, and the animal stayed onto a platform, isolated platform, where it's safe, and when it goes down with the 4 paws onto the grid, it got a mild food shock, and then it is returned to the home cage. And what we quantify were the training latency, which is the time the animal takes to go down onto the grid, and the test latency sometime later, where you quantify the time to get down onto the grid. And if it is 24 hours later, for instance, you can assess LTM, long-term memory. Well, the latencies increase. The orange are the test latencies, and the yellow one are the training latencies. And as you can see here, the latencies are higher as the food shock is stronger. And the threshold is about 0.5 milliamperes. This is the intensity we use in most of our studies. And how we can dissect the regions? Well, you listen to the nice talk by Beatrice, where they study the histaminergic pathways and receptors in memory. And in general, what we have to do is a stereotaxic surgery, a delicate surgery, very precise, put cannulae onto the brain, into the skull, going to the region we want to impact. This is the dorsal hippocampus. It's just a drawing of a section of a brain section. And then in the awake animal, you can inject the drug, which is, well, actually something that impacts on the receptors of the transmitter system you want to study. A relevant concept that impact in our understanding of the relationship between brain and behavior surged at the end of the 19th century with Claude Bernard in France, Paul Ehrlich in Germany, and John Langley in England, who proposed that drugs do not interact arbitrarily with cells, but binding specific receptors localizing the cell membrane, exposing the surface. And this is the fundament of the physicochemical basis of the intercommunication between nerve cells and of the drug's action onto the nervous system and on every tissue. And you know that the central nervous system consists in interconnected neurons that are connected by synapses, but each cell is independent. And this was clearly shown by Ramon y Cajal, who proposed that the nervous tissue is a network of interconnected nerve cells, but not but individual cells, independent cells, using the argentic techniques of Camillo Golgi. And as Candel drew in his beautiful book, nerve cells, it's to say neurons, are the main signaling unit in the nervous system. And they are in general like this, with a receptive region, which we call dendrites, or including the membrane of the somata, and another projection, which is the axon. And by the axon, the impulse, nerve impulse, or what we register as recorded as the action potential, travels and arrives to the end of the axon, which we call the axon terminals, that are the presynaptic side of the contact of the synapse, and then contact with the next neuron, which is the postsynaptic neuron, and in the receptive side of the postsynaptic neuron. There are 100 billions of neurons in our brain with a similar basic architecture. Then a key to understand brain organization and its emergent properties as cognitive functions like learning and memory, we have to understand that neurons with similar properties can produce very different actions based on the way they are connected, how and whom. And to dissect this, as I said before, to individualize which neurotransmitter or system we want to study, we have to select the sort of circuit synapses we are going to impact. The neurotransmitter is released at the, I have some signal in my sound. I don't know if you want to say something, but I continue. The neurotransmitter is released from the presynaptic side contained in the synaptic vesicles to the cleft, synaptic cleft, and impact on the receptors in the postsynaptic side. And these are specific for each neurotransmitter. in this case, you have the terminal, axonic terminal, that could release the neurotransmitter and impact into the neuroreceptors, but it's possible to use an antagonist that block this synapse. And what we are doing in general is to use antagonists, but you can also use agonists or modulators. From 1989 to 2011, Ivan Izquierdo, together with Jorge Medina, another Argentinian investigator and close friend of mine, and about 20 other colleagues, published more than 200 scientific papers making remarkable discoveries. In review, they publish lots of biochemistry and pharmacology of memory. And there was a brief paper in Nature in '98 about the independency of LTM, long-term memory, from short-term memory, showing that the blockade of short-term memory is not a condition for impeding or having amnesia in long-term memory. And another remarkable discovery was a new phase of longest memory consolidation, now named memory persistence. Ivan also collaborate with many of us, and that work generated an enormous amount of new information on the role of neuroreceptors, second messengers, gene expression, and much more involved in memory processing. Here is Jorge Medina on the left, Beatriz Ferreira, which also participated in those times, and Ivan Izquierdo with Yvonne, as you already know. Well, the important thing was that we arrived with these tools to understand some important things in the synaptic neural integration on memory consolidation. Just a cartoon to show that on a postsynaptic neuron, you have excitatory entries, presynaptic neurons that release, well, 2/3 in the hippocampus release glutamate, which is an excitatory amino acid. Then most of them are glutamatergic synapses, and also have inhibitory presynaptic entries that release GABA. GABA is gamma aminobutyric acid, which is inhibitory. 1/3 of the neurons in the brain release GABA, are GABAergic. Then with excitatory synapses, you have the possibility of produce excitatory postsynaptic potentials, and with the inhibitory entries, you have the possibility of develop inhibitory postsynaptic potentials. As these signals are graduated, are not or none, as are the action potentials in the axon. Then in the axonal cone, all those signals could integrate, and an action potential or nerve impulse is released. And at the graduated signal level, at this level of the synapses, where the synapses excitatory entries or inhibitory entries are acting, the modulation of memory could take place.
1 minute now.
Okay, thank you. I will go quickly. Then in studies about benzodiazepine GABA neurotransmission, we arrived to the conclusion that a benzodiazepine receptor-mediated endogenous mechanism is activated during learning, and that this blockade facilitates retention of information. I will leave this, and we made a special study, complicated study on 3 different regions, which are the medial septum, the amygdala, and the dorsal hippocampus. And without going into details, just here, you have the representation of the inhibitory avoidance task, the controls, and in each of the regions I said, amygdala, medial septum, and the hippocampus, with antagonists of acetylcholine muscarinic system and of glutamate transmission and noradrenergic transmission, you have amnesia. And on the right, with the agonist, we have facilitation of memory. Then this was the drawing by Ivan. I want to show you. We could conclude that similar synaptic mechanisms in the amygdala, medial septum, and the hippocampus are involved in memory consolidation. NMDA, which is glutamatergic, muscarinic, which is acetylcholine dependent, and alpha noradrenergic receptors stimulate, and GABA receptors inhibit this process, and alpha noradrenergic receptors modulate the GABAergic synapses. In the inhibitory avoidance task I show you, these mechanisms operate in the 3 structures, in amygdala, medial septum, and hippocampus, while in habituation, only those in the hippocampus are operative in the open field. In each structure, these mechanisms would regulate a different aspect, component, or form of memory. Then both inhibitory and excitatory neurotransmission are involved in memory formation, particularly in memory consolidation. Well, I will go to the end of the pictures. We'll skip all this. And we, together with George Gilfeld in the Urgis University, show that NMDA receptors, glutamate receptors, are involved in consolidation of memory. And it was shown that the blockade in acquisition and also reconsolidation. The main conclusions and achievements from the studies I show you are that benzodiazepine GABA synapses in the hippocampus modulate memory of habituation. Similar synapses in septum, amygdala, and hippocampus regulate memory of avoidance learning. Through localized microinjection of specific receptor agonist and antagonist, GABA synapses inhibit cells excited by glutamate via NMDA receptors and by acetylcholine via muscarinic receptors, both necessary for post-training memory processing. Noradrenergic synapses play a modulatory role. Neurotransmitter receptor interactions occur in the 3 structures I mentioned for inhibitory avoidance, but only in the hippocampus in the case of habituation. And there are 3 separate mechanisms, each involved in the regulation of different aspects or components of memory, all with a similar synaptic arrangement, and all done regulated by benzodiazepine GABA receptors, probably in response to anxiety levels. The participation of different neurotransmitters and neuromodulators in memory processing, from GABA and benzodiazepines to glutamate and acetylcholine, have been studied, unveiling their inhibitory, amnestic, or facilitatory effects respectively, which could be implicated in healthy as well as in pathological conditions. And these are the people who make the work. And this is a meeting of our lab with colleagues like Georgie Kilfeld in the center, Carla Dalmas and Randri Lee Mescher, and also Martina B from our lab, who was not in the first picture. And thanks, obrigado, gracias, caro mestre.
Thank you, Diana, for the nice presentation. And I would like to move now to our friend, dear friend, Carlos Alexandre Neto, que é professor titular do Departamento de Bioquímica do Instituto de Ciências Básicas.
Of Biochemistry of Basic Science at the Federal University of Rio Grande do Sul, that is going to talk of another aspect of Dr. Izquierdo on the universe of memory, which is the role of the hippocampus, the novelty and memory. Hello? Hello? Alex, can I see you? Good afternoon, Esper. I would like to thank you for having the opportunity to contribute in the homage to Dr. Izquierdo, that contributed so much to the neuroscience and to the memory. I would also to thank for having the opportunity of sharing this moment with Dr. Esper and Diana. We worked, I wouldn't say together, but we were contemporary in some labs. And with Beatrice, that it was a recent acquisition that I just met very recently during our tribute. So I will talk a little bit about the hippocampus, news and memory. This is part of Dr. Izquierdo's activity that I followed closely. And then I also contributed for this development. The hippocampus is a structure that is here, is deep inside, and is important bilateral structure in the mammalian brain. And Diana and Dr. Beatrice showed you the hippocampus in rodents. And the case of ideas, the hippocampus, it came into fashion in its real relationship. And when we had this patient HM Canadian that suffered a bilateral ablation of the hippocampus, and because he suffered epileptical crisis that couldn't be treated with drugs. He did not have any more epilepsy, but he developed severe anterograde amnesia. So during his 40s, and we learned about, and from this time we learned about. In the 70s, we had evidence that rodents and non-human primates, the hippocampus is also very important for the memory function. This was compiled in a very important book. This is a pillar then that Izquierdo studied profoundly. The other pillar is the issue of the opioid and the function memory. Professor Izquierdo was all a pioneer in this field. He developed pharmacological studies showing that opioids could cause amnesia. At the time we used naloxone. This after the openness and antagonist could facilitate the memory. This is because of the agonist that amorphine that causes amnesia. On the contrary, the other ones can cause new memory. In the beginning of the 80s, we already showed that neurons and the brain produce endorphins and encephalins. And this, we know, Izquierdo was the pioneer in the world to study the role of opioids endogenous in the memory. And this also was also sustained by the discovery of opioids in the neurons in the brain. I would like to remind you that the Nobel Prize for 1977 was for the demonstration for the signaling of peptides in the brain and the immunoassays for detection. And the two awarded were and these were the fundamentals for the Izquierdo studies. The fundamental question: memories can be regulated by endorphin. Why did Izquierdo make himself this question? Because in the years 82, 83, 84, he was studying the amount of beta endorphin in the brain. In the middle of the brain that contains the hypothalamus and more of genetic tissue of and he showed that the endorphin when animals were taught, but this was not clear if they were taught only by learning or stress, or this fundamental study that they made in 84, the first test session which I participated, we tested several forms of behavior, always learning and memory. And what we noticed, and now I want to show you this chart, but we saw that the endorphin was always lower when there was a new situation, independently if this new situation represented a situation of learning or stress. Therefore, the beta endorphinic system is activated by novelty. The lowering of levels was detected by interaction. And the second scientific question, and here we could see we see the hippocampus is receives several calls, and by our previous projections, and we have the collaboration of our friend and coordinator Esper Cavalheiro that developed these techniques in order to support all these news. So we isolated the diencephalus from the different fronts, and we tested if the beta endorphin was still liberated in a novelty situation. This slide showed that the lesions, the pre, for, and before had no effect, reinforcing the role, the idea developed by Jeffrey Grace that the hippocampus detects memory and works as comparative. And when he shuts off the comparatives, it's the one that defines if there is novelty or no. And the atmosphere that we are living, if it doesn't work and it doesn't send the message, there is no interaction with the memory. Here, here we have a slide for my picture to show you how young we were in the beginning of the 80s. So therefore, what was the idea, the key idea that Izquierdo, that beta endorphin is liberated because if the animal detects has something new, there is nothing in its memory that can solve his present situation. So he has to be alert and to see how he can solve it. So the memories will be temporarily available because of the beta endorphin that is liberated metaphorologically. After this, our master evolved to other studies, mainly for, and I went to do my PhD to work with Jeffrey Gray in hippocampus and brain ischemia. Here I have a memoir for a conference that was in Magdeburg in 1989, where we were living at the time when Izquierdo came there, and 1 month before our trip, the Berlin Wall came down. So you see, we are exactly standing while the wall. But anyways, it was marvelous. We profited so much of this event. This moment marks exactly the moment of my independence, but he remained my mentor, and I started studying this brain ischemy. But Izquierdo continued working on novelty. If there is a liberation of beta endorphin after the liberation of novelty, after the task, this is not new anymore. But if I give him some kind of opioid before the test action, I improve a lot the recovery of the memory. I have the evocation, but the beta endorphin works before the test session. Maybe if I put this in a novelty session, showing that the novelty improves a lot the memory of animals. This is very interesting because this is a concept that I can use this as a transactional concept in human beings and submit patients and to measure the capacity of retention of memories. And this can improve, and this can be used in the treatment, in the daily life, in the studies of memory like dementia. But the paper that I brought here was of the year 2000, showed that this facilitation of the memory by novelty depends on the hippocampus. In this article here, Professor Izquierdo, they blocked temporarily pharmacologically the hippocampus, and with this blockage, the novelty lost its facilitating effect in the memory. The novelty facilitates something that Beatrice mentioned at the beginning, that is the extension of fear. Let's review very quickly. There's a test that you see that the animal is put in a box, in a platform, and he's going to explore. He doesn't know what's going to happen, so he has shocks. 24 hours. I test again. I see, well, he's not going to go down to the grid again, but eventually he goes down again. If he goes down and does not suffers any kind of shocks, and the next time that he's going to be put there, he's going to go down quicker, faster. Then this is what we call extinction learning. The animal learns that this stimulus, painful stimulus, previous stimulus is not there present anymore, and he can explore his environment. If I make a session of novelties before extinction section, the animal will have a very faster extinction. He's going to go down much faster than the other one. This is very important. Yes, this is very, very important because this involves the hippocampus and the family of hippocampus, and we know that dopamine is a transmitter in various roles in several pathologies. Also very important because if I learn to extinguish fear in a determined situation, this can be interesting if I understand this mechanism, because it can use for the treatment of stress, of post-traumatic stress, very frequent, that impairs a patient when he has too many sessions. Here on your left, something that Professor Izquierdo worked since the 90s, or 95, 96. He was also very, very interested in the molecular modulators of the memory. This is a study on extinction, on the facilitation of the extinction of the fear, and showing you pathways, hippocampus pathways, evolving not only the glutamato sectors and the nervous system, but also and the signal reaches the nucleus to a phosphorized path that is a substance that works releasing the DNA. I would like to remind you that the Nobel Prize in the year 2000 for medicine was for Dr. Condell, that dissected the molecular pathways of the memory processing in molecules, and our master Ivan made for a long time the dissection of those vias, this dissection vias in Reuters. Our great master also worked for environmental enrichment. This means to face challenges and novelties where rats live every day. And he demonstrated that this evolves. The enrichment environment reduces the deficit of memory in Alzheimer's patients. Reminding of Dr. Diana said previously, we have here a slide, a sketch made by Santiago Ramon Carrar of a section of a stimulated brain, if a stimulated animal, of an animal that used to live in an open, and from another animal that the first one was lived outdoors, and the one that lived in a cage and indoors. You can see the difference of synapses and neurons. The environment enrichment leads to a experience-dependent plasticity. We also work in this field in the role of prenatal ischemia that involves the hippocampus. I'm not going to talk about this. I only would like to say you that this environment enrichment. So the legacy of Professor Izquierdo, besides all the ideas that all three of us here highlighted here, Professor Izquierdo published more than 600 papers. He has more than his 25,000 citation. He supervised 1,000 PhDs, doctorates, and dozens of postdoctorates. He influenced five generations of neuroscientists, and he had a unique contribution for the advancement of science, especially in Brazil and Argentina. He left us a fantastic legacy in terms of science, his ideas. He was a brilliant scientist, intellectual, generous friend, a very generous friend. And I would like to tell you a couple of stories, but unfortunately we don't have time. A family man, a great father, grandfather, and husband. And we only have unique memories from here and very inspiring. So my thank you very much to our masters. And I would like to say that today that I developed all my career developed based on everything that I learned and that I worked together with Dr. Izquierdo and all the colleagues at the department. Thank you very much. Thank you, Alex, for your excellent presentation. And I say that I was very excited listening to you. A long time I haven't dealt with the memory, and this was a great opportunity to review the absolute beautiful life of Professor Izquierdo, and how many people he touched with his charisma and his willingness to help. During his career, he worked with what he had at his best. He had a fantastic memory, saying to all of us. Beatrice, I hope that you understand everything that I'm saying. I know I'm speaking quickly, and I'll try to go slow. He was always able to show us in the book and the exact page on the right side, the exact sentence that he wanted to remind you, and he knew this by heart. And he always said this, and he always used to say that novelty is what leads us, what makes life, and novelty is what makes us thrive. I'm going to see if we have any urgent questions from our public. And I would like to make a couple of comments on novelties, for instance, porque a pergunta,
because going from the memory, the pathways, and all the possibility of having memory, extinguishing memories, and so on, and the role of novelty. Remembering my personal life concerning the things that we do during, is that the duration of time according memory. I'd like to know if there is any kind of a circuitry, because it's very clear that if you are living a lot of novelty, when, for example, you are starting a new travel, are going to a place that you've never been before, the time almost disappears from you. You do a lot of things and you think that, okay, and you remember everything. When you do a lot of routine work, the day is very long. It never ends. The day is extremely long. And you say, what I've been doing today, you don't have a memory what happened to you. This is really interesting. And I'd like to know if you have thought about the possibility to have the time circuitry in the brain, because you have a very good estimation of the time, and the possibility of remembering things or forgetting things, of course we remember what touched our heart. We can remember easier what is very close to us. And I'd like to know about from you. You can answer or think with me about the possibilities to join the things between time, circuitry, memory, novelty, and so on. Go ahead.
May I say something?
Yes.
Well, from the experimental point of view, just with the Ratas model, we published some papers on the effect of novelty on the amnesia provoked pharmacologically by, for instance, scopolamine. If you block the muscarinic cholinergic receptors, then it's to say, if you block the cholinergic system to the hippocampus, for instance, you could overcome this amnesia by novelty. If you expose previously the animal to novelty or to another environment, for instance, you can overcome the amnesia caused by scopolamine. This was astonishing for us. And then we continue studying this. What I mean is that what Carlos Alexandre studied was extremely interesting and with a very, very high translational value.
Ok, ok.
Esper, essa questão do tempo, ela é uma questão muito...
This issue of time is very interesting.
Nós sabemos que
we know the hippocampus has a very important role in the circuits of time controlling, and we know by studies in human beings, for instance, that with musicians, maestros mainly, that play and they conduct by memory all this huge music, and and they do all this time correctly. And when this is compared, they do this by heart and compared when they are in front. So the hippocampus has very specialized cells, the placement cells that Joe Kiffins that demonstrated, and that the researchers and the Danish researchers researched and got the Nobel Prize. But I would like to say that the novelty induces a change in the activity of the hippocampus and directs the activity of the hippocampus while it's there present. The memory can either be affected or conducted by an important way by the hippocampus, because the news of the hippocampus makes it become more alert, and we pay more attention on what is surrounding us because of the situation of novelty, different when you have already a routine. And in the same way, when you have a situation of emotion, it's going to be the amygdala that is going to command. So it will have at least the participation of those very important structures that Diana and Beatriz mentioned from other areas. But your question shows us in evidence and practical evidence the importance of the modulation of those two topics. Thank you very much, Alex. Eu gostaria de acrescentar que
yes, of course, the hippocampus is a sort of the organizer of many memories. And I would like to add also, I am biased, I know, but the histaminergic system and the noradrenergic system are, I think, very important. I remember Jim McGaugh was the one who in the 1980s was the first one to realize that the noradrenergic system gives the right amount of, let's say, alertness to the whole brain in order to activate the brain and consolidate memory. But if you have too much of it, you have the opposite effect. So you have a inverted U-shaped curve. And I think that the histaminergic system somehow is also important in switching on at the appropriate moment and for the amount of activation, like novelty, as Carlos was mentioning, and turning on, for instance, the hippocampus for the animal to recognize the place where it found the food. And so that's a novelty, and it's an important stimulus for the animal. He knows that she knows that under that tree there is good forage, and under the other tree there is the snake that is going to bite you and kill you. And I believe that among other circuitry, of course, and I have been working on the histaminergic system for a long time. It's the histaminergic system is one of the brain structures that tells the animal, okay, amygdala, switch on. Here is the novelty of the snake. Remember this, send this memory to the cortex and fix it there. And then the animal goes to the other tree where there is the good forage, and probably the histaminergic system helps the hippocampus say, well, okay, my place cells have to remember this tree, this particular environment, to go back and forage the next day. So this is my bias, of course, but maybe it's a sort of a reasonable guess. And I think that we need so much more research to either validate or dispute my theory.
Great. I think it's a very good possibility to understand what, but there is a very huge pathway still missing, that we need to follow to understand. Now, in recent years, I think that the psychiatrists start using, or behavioral psychologists start using the extinction of memory to treat several kind of diseases linked to bad memories or events in past life. I think that how this is clinically progressing these days. Do you have any idea, Beatriz? How is this really helping people? I know that there is a lot of papers on this, but I'm not following this literature particularly. But we know that they are using several techniques to avoid this memory. Probably histamine is always involved in this.
Yeah, well, from the psychological and psychiatric point of view, there are several techniques to help people affected by the post-traumatic stress disorders. The most popular is exposure therapy, and that consists of re-exposing the patient to a threatening, like photographs or stimuli that bring back the memory, and then work. Well, what has been used so far is the propranolol or a beta antagonist, which, as you may know, has several side effects. Propranolol is not even used anymore in therapy, just for, I think, for essential tremor, but it's not an antidepressant any longer. So, and the success is limited. There are also other techniques, and I don't remember the technical name, but the subject sits through several therapy days, and it consists of focusing the attention on something that moves around your pupil. So I really forgot how the technique works and what the name of the technique is. Other psychiatrists have used arts, and so letting the patient describe with painting or creating images that evoke the frightening situation. Antidepressants are being used, and but as far as the histaminergic system is still completely unexplored. As you know, probably the limitation is that the histaminergic compounds that we have available, they cause drowsiness and sleepiness, those that pass the blood-brain barrier, the H1 receptor antagonist. H3 antagonists, they wake you up, and patients that are treated for narcolepsy take advantage of the waking properties of the H3 antagonist, but I'm not sure this would be useful for treating obsessive compulsive patients. So from the pharmacological point of view, I think that the histaminergic field has to be really explored with new compounds, and would be nice to have to deliver the H1 receptor antagonist only in the hippocampus, but of course you cannot do that in humans. And so I think we have a lot of work to do in this respect.
Okay, our president is raising his hand, so I'll give the space for him to do a question for you. Professor Luiz, go ahead.
Okay, bom, obrigado, thank you. Primeiramente, eu quero cumprimentar todos vocês aí pela participação nesse webinário.
I would like to greet you, all of you, for participating in this webinar, for this tribute for Professor Izquierdo. I would like to make you a laic question, maybe something that is in other people. We are being submitted each time more to a massive wave, say maybe information of disposition of internet, TV. Children also are being submitted to all this new wave, studying through internet, and all of them, you know, spend their day on the iPad. So I ask myself, what are the social implications, neurophysiological implications, especially on the memory? Because recalling the work of Dr. Izquierdo, because learning is similar, works together with the extinction of memory in order to absorb novelties. But maybe is the big news here now, and I don't know how our brain is adapting, and how the child's brain adapts and being submitted to this kind of information. Do you have any comment on this issue, please?
If
Eu gostaria de fazer um comentário.
So you're really right. This is becoming another pandemic. The addiction to internet, addiction to the games, the telephone or games. Now, I think that in the new DMS manual for the mental diseases, it is really classified as a new kind of addiction. So which is really threatening and I think dangerous, especially in children. I have really no idea what the perturbation are in the brain. I have not really addressed this issue. Probably some of my friends know more about that, but it's certainly becoming a really classified psychological problem that of like drug addiction. This is a addiction to the internet and to the games, like what we call ludopathy, which is the addiction to playing and gambling, to gambling. So, but I have no idea how and if and how psychiatrists and psychologists are facing this problem and taking measures to treat these patients. I don't even know where these patients are aware of their problem and whether they ask psychologists or psychiatrists to be treated. So I don't know if I answered part of your question, because of course I don't know more than that.
Tem alguns estudos já vinham mostrando que as crianças
showed us that children are spending a lot of time in computers, and they do not develop their manual skills that involve three dimensions, and they are, you know, the joke, the things kids play, how they used to play before. This was measured before, but these are studies that will take another, but this was 10 years ago. Our brain adapts itself very easily to any situation. We have a huge plasticity that depends of the stimulus that we receive. So even in a situation that wouldn't be, let's say, quite the desirable, like the excess of the use, we adapt ourselves and retrieve the best we can. But as Beatrice said, but we have people that become addicted and they have a period of abstinence, and this creates a problem. Maybe this, and we should look for a treatment. But with the pandemics, but before the pandemics, we thought of this as a problem for the future. This became a problem from the present, because all of us and all the children, we spend all of our time, we are stuck to a screen, at least for 8, 9, even 10 hours a day. But I understand that maybe this can have serious consequences, but our capacity of adaptation will be even bigger than these consequences. Of course, not mentioning the issue of abuse. When you have a pathological behavior, of course, you need treatment.
Posso falar?
Pode, deve.
I would like to.
I have not studied that, but I read some things, and also I have a niece that is a psychoanalyst and published some books on the subject. If you want, then I could send you the details. But this is very interesting point, because it's completely different from the children and us, because we already have another sort of relationship with people and with the other ways of information. But children, probably from the last 10 years or 15 years, are already born with this, and probably they have some skills, as Esper and Alexandre said. It's interesting because some people studied the difference in the brain behavior when you write by hand and when you only write by computer, or it's not just the type of machine. We are always writing in the computer, and probably we, the oldest, write by hand, but the young people probably don't write by hand. And the thing is that when you write by hand, the thoughts are more or less in the same, or are faster than your writing. But when you write in the computer, probably it's not the same thing. And this is very significant at the level of synapses, and probably at the level of the pattern of activity in the brain. What was the thing that I read was studied. The patterns are different, and then probably we have different development of circuits in children than in the adult like us that are hybrids. Okay.
But yes, but maybe this is not necessarily bad. They are evolving in a different way, and maybe in 100 years we will never use a pen anymore, and maybe our fingers will start to retreat, and
Yeah.
so we will not eat our hands anymore. Maybe the evolution is working this way. Who knows?
Yeah.
They have other skills. I see, for instance, children, they can handle a telephone, the computer, and listen to the teacher. So maybe they have a split attention, and I really don't know. Or maybe it's
No, we don't know. We don't know. The multitasking effect of this
which is
could
something that we're
be.
not good at. At least I am not good at splitting my brain between different tasks. So and maybe they are evolving a different way. They are acquiring new skills and dropping those that are not used anymore in a sort of a Lamarckian way.
Yeah.
But as you said, this is something different in more or less normal people, if we can say normal people, and in psychiatric syndromes or illness, and this was the thing my niece was studying, and because she works with children with problems, with neurological problems or genetic problems, and this is interesting because it's different when there is a psychiatric entity there.
Sure.
Eu gostaria agora de passar algumas perguntas que estão chegando através daquelas pessoas que
Would like now we have here a couple of questions. We have a couple of questions from our audience. I have a question directed to Beatriz from Bernardo Santinapi, an Italian fellow, that is asking como os
with the hippocampus in order to access memory data.
Well, why do you need to answer this question?
Por que eu preciso responder essa pergunta? Em termos de o hipotálamo, que está ligado aos neurônios, manda poucas projeções para o hippocampus. O hippocampus está carregado de receptores. O sistema estaminérgico e o jeito especial que isso é transmitido, isso é liberado através de veias pelo caminho e
some histaminergic neurons that project to the hippocampus release histamine. Histamine binds to postsynaptic receptors H1 and H2, And these have been found to excite the postsynaptic cells. So we really have no idea which neurons in the hippocampus that store or momentarily or definitely a type of memory is excited by which neurons. But we believe that when the histaminergic system is activated in the hippocampus, it's the histamine is being released along the dendrite, and the neurons that become in contact, the receptors, the postsynaptic receptors are excitatory. So the hippocampal cells, the pyramid, if it is a pyramidal cells, it becomes excited, and maybe that's the neuron that holds the trace of a particular memory, or it may activate the GABAergic receptors, as Diana was saying before. GABA is the most common and abundant neurotransmitter in the brain. And it is the sort of the fine regulator of the excitation in the hippocampus. So maybe the histaminergic neurons affect that particular circuit by balancing the inhibitory GABAergic release and the glutamatergic from the pyramidal cells, and you probably reactivate that particular circuit that is necessary for the animal to remember. There are ways to address this question, and by using optogenetics and or other pharmacological the chemopharmacology, the new techniques that allow you to selectively shining light on mutated cells in the animal. You can selectively activate only those cells, for instance, that express histamine or that express glutamate. So this has been done by Tonegawa in Japan. I think he was the leader of these new techniques, and he actually managed to reactivate memories. And so the animals expressed fear that it had learned in one environment, and then you place the animal in the environment B, which is totally neutral. And by reactivating those particular neurons, the animal freezes or expresses a behavior that is typical of fear memory. So maybe we can transport this technique and have the same thing being done with the histaminergic neurons. But actually, this is something we're working on. So maybe in 5 or 6 years, I can give you a part of the answer, let's say. I hope so.
Os outros gostariam de colaborar na resposta? Tem uma visão concordante? Não? Está tudo bem? Posso passar?
Anybody else would like to add something? Here we have another question from Marco Tulio Rodriguez. This question is for all the 3 speakers, and he would like to know por quê
to treat the post-traumatic stress disease, or comment about the relation between psychedelic drugs and memory. Any of you would like to?
I'm trying to remember psilocybin.
Yeah.
I think it is being tried as an antidepressant. Correct me if I'm right.
Yes, but was abandoned. All of these psychedelic drugs were used in treatment in some time, and all of them were abandoned.
So they are exploring the psychedelic drugs for the treatment of depression, but I know nothing about PTSD. So I give up here. I don't know more than this.
Os estudos, eles existem estudos, eu não saberia.
Studies. I wouldn't like to know. I can't name exactly which one, but the effects depend of the kind on the receptor, on the kind of receptor where these drugs act. Originally, this idea started with tests using LSD in the 70s, 80s. And this was, they were really taught in order to cure affective disorders. But in the end, it's not useful because these are drugs, these are substances that produce abuse, that lead to abuse. And the pendulum between the positive pharmacological effect and the abuse of substance, it's very complicated to use. Maybe what can be, maybe where we can, an application, a more close application would be the use of the derivatives of cannabidiol, because yes, there we have with some effects, very punctual effects, including on memory, and that eventually could be tested in humans, in human beings, in the same way. And its effects, anti-epileptical effects now, has been recommended to treat some kind of refractive epilepsy. I would say that we have a long way ahead of us for the development of this psychopharmacology, but this is a fascinating theme.
Pois não, Diana, pode falar.
E só para completar, além disso,
besides,
they have lots of adverse effects, collateral effects, and then were not recommended to use in humans. But as Alex said, the cannabinoids are now in strong developing and many clinical trials and so on. And I guess this is probably a good thing.
Yeah,
Muito bem. Go ahead.
If you can comment, let's not forget the cannabinoids can trigger psychosis in some patients. So it's always tricky, and I think that the anamnesis of the patient has to be really very, very clear before you use the cannabinoids, as there are cases of really important psychosis that have been triggered by the use of cannabinoids. This is the daily chronicle of children that end up in the hospital because of recreational use of marijuana and cannabis, and then they end up in a hospital ward because of a really serious psychosis.
Yes, apart, you know, there are works in the line of Ivan work by George Kielfeld, and also people here in Argentina, in Cordoba, in cannabinoids and memory. And it's interesting that what Beatrice said, because they have some important effects on memory also.
Esper, teu microfone está mudo.
Ok. Já corrigi, eu percebi rápido. Nós temos mais perguntas, e para não deixar as pessoas um pouco sem resposta, mesmo que eu possa depois mandar para vocês as perguntas que ficaram sem resposta, mas tem duas aqui que eu vou fazer juntos, que são mais ou menos referentes ao uso de computadores. Uma é da Patrícia Fernanda Schuch. Ela diz que atualmente os alunos, normally these days the students, they go to the classroom carrying a computer. Then what we used to use is, I mean, paper and pen or pencil. She's just asking the fact that to use the computer to digitate instead of writing will be interfering with the process of memory and learning. What do you think about? And the other question is coming from Elba Venancio. She is just asking you if you know how to moderate the excessive use of tablets, iPads in classrooms. I don't think that we will be able to. I never been able to say, I just close your computer, stop using it, you know, but this is not a polite way of using my authorship, my authority as a teacher, but I think it's very bad. But if you'd like to comment these two questions, I thank you. And then Hernani is just commenting your answer to him and asking some other, giving another, is not question. I think I send it to you because we should close our talk here, but I will leave you with these two questions and then we see if there is more time to go ahead. Do you remember the questions about the use of? Okay.
I could comment on the first. I guess from the studies I read and from the experience that it probably is important in the alphabetization period that the kids learn how to write by hand. But once it is complete or it occurs, nobody knows about, as Beatrice said, could be better or faster to write by computer. And actually now most of us write most of the time on the computer. But I know that, for instance, my grandson in the school, we use a tablet, don't use a book, or most of the time they use a tablet. And this is okay if they are rightly guided to find the things they have to find and read the things they have to read and learn what they need to learn. But the thing is that in the alphabetization period, when they are very young, the kids, probably we need this to form some sort of circuits and synapses that are dependent on this relationship of the brain with the hands and with the skill, this skill, and also with representation of language. And while this is, we don't have experience enough in the human beings, but probably it is very interesting to study this, and some people is studying this.
Yes, if I may add a comment, this is, I guess, I think is a totally new world. And I think it has been accelerated by the pandemic. I think the children should be warned against using the telephone in class, in school. That I think is a distracting, really distracting and inappropriate behavior. But maybe you're right, Diana. Maybe in the future we'll only use tablets once we have learned how to write, and maybe we will emphasize the reading, which I think is absolutely irreplaceable for our mental and cultural development. So I am not in the position of giving advices because I teach to 20-plus-year-old kids. And there is no way you can change their behavior. So they're on their own. And if they want to listen, they listen to the lesson. If they want to play with the PlayStation, there is not much I can do about it. But I think that certainly children have to avoid using the telephone in class. And I know that teachers somewhere ask their children to deposit the telephone on the teacher's desk. And then when they are in recess or leave the class, they can take their phone and communicate with their friends and parents. But during the lesson, communicating with the computer is prohibited.
Eu gostaria de fazer comentário também. Nós sabemos que a memória...
We do know that memory depends fundamentally on attention that we deposit at the moment that we are living, and I think that it has to be later on remembered and evoked. And something very difficult is to pay attention during a very long time, hours, facing only one stimulus. For instance, the screen in your computer. So the act of being navigating and looking at several sites doesn't mean necessarily that this person, in the case of child, that she is actually learning everything that she went through. She's making several movements, several clicks. This is intuitive, but necessarily she's paying attention. And maybe we don't know if this is going to become a relevant piece of information. This issue of attention is very important in the learning process, considering the use of the computer or the screens or the cell. But even in presential class, face-to-face class, where the teacher, is doing his best in order to keep the attention of his class and follow his thoughts. So this issue on attention is fundamental, and I think that the more important thing, besides the fact of using more or less the resources of technology. Another thing that I would like to add, not only for children, but also for the children, is the source of information. Many people think somebody that has a cell phone, a laptop, or a computer has all the available information. No, it's not true, because on the internet we have all the available information, and another thing of trash that is transmitted as information. Therefore, I think that this is an issue very important and serious point, because internet is a tool that allows us to introduce to everything that's being shared, socialized. The only problem is that very big share of this of being shared is trash, crap. We have a source. We have trustable sources. And this is something that when we talk about remote learning and how we are doing during pandemics, that I think this is a practice that is here to stay, as well as the remote work is establishing itself all over the world. And I think that even pandemic, the remote learning is here to stay, and we'll have to work. We teacher, we will have to work in a hybrid way. But I even think that in elementary and high school will be working the same way. This is the way we are exposed, and we will have to deal with this cerebral brain plasticity. I agree completely.
I want to say, apart from that, in 2 weeks, I understood there is an important meeting there about algorithms and artificial intelligence. And I want to add this to Carlos Alexandre, that we not only got information and lots of useful things, but also there are dangerous information, dangerous things, because there are not only people, just enterprise dedicated to create algorithms, but for insert false, fake news, false information. Then this is extremely complicated, and I guess that the systems that drive millions of data and have developed algorithms to use this information to in some way direct the results or your mind, are also something important, especially for kids, that we have to take care of this and to avoid this, because it's too difficult to avoid this.
Okay, just to close, I'd like to be fair and read the comments by Hernani Fantinati. If you then would like to comment very quickly, I read it for you. That is concerning the idea of reaching the neurotransmitters, reaching or signaling the hippocampus. It says that comparing it to the artificial, as you were talking about, intelligence, is kind of interesting because you use an activation function to let pass or not pass the input signal, which is neurotransmitter from the biological neuron. Regarding the learning process, I would understand that it looks like, sorry, because my eyes are very bad now, you know, it looks like weight compensation, which in the biological perspective could be the neurotransmitter type. I think it's, at first I would like to suggest to Hernani also come to this special event of the Brazilian Academy of Science, talking about artificial intelligence and algorithm. I think it's very close what we are talking and discussing here. We don't know, you know, the brain should have a lot of algorithms on its own. And our task is just to discover which algorithms the brain is using to understand the world, to use it in our thoughts and emotions that we think. I'd like you to comment, guys, on this comments from Hernani, if you have any. Otherwise, okay, because we need to close, it's almost 6 o'clock and our time is finishing here. Comments from you?
One comment, only one comment, which is, yes, we need many algorithms for understand our brain. We have 100 billion of neurons with more than a dozen, in between a dozen and 10,000 synapses on each neuron. Then, well, imagine the huge amount of data. It's a big data enterprise, and sure, we need to understand more this, but the difference with any artificial intelligence you want to use up to now is emotion, because you have like a very, very important load in our memory, emotion. If you don't have emotion and attention, as Alex said, you have difficult problems in storage of memory. Then to storage information, to keep information or representation, which is the proper name, we need something that is like weighing that with emotion, or emotion and attention, or with attention. Okay.
Yes, if I may add a few words, you're absolutely right, Diana. And let's remember that also perception. We know very little what perception is. You know how the stimuli from the environment reach our brain, but then what makes you feel, or what makes you think that you have a brain? I think this is the only organ in our body that thinks of itself. The liver does not think of, as I imagine, does not think of itself as a liver. So now, you know, there is the machine learning, the deep learning is the new venue for trying to figure out how our brain works. So they have terabytes of information that are fed into a computer, and the operator chooses the algorithm to make the machine learn and react. Robotics, I think, is based on some kind of machine learning and deep learning. But I don't think you can make a brain in a dish or with the help of electrodes and computer and algorithms. So we're still very peculiar, and I doubt we will ever see a brain working as an artificial brain, although, I mean, progresses are being made with machine learning, with these very complicated, very complex algorithms that the engineers informatics are elaborating.
Também adicionar comentário, os modelos matemáticos.
Add that the mathematical roles that are available today, either for machine learning or robotics, they are still they are simple machines compared to the human brain. If we focus on the hippocampus, that is a very well organized structure and very well known, what we see is that many receptors, many neurotransmitters, and many neurons doesn't transmit before synaptic. This means complexity that at the moment, in mathematical terms, I'm not able to put this in my algorithm because my algorithm needs yes or no. Therefore, I think that is very important to continue studying and raising the complexity of models, of roles in the studies. But I think that we are still sometime far of the precise learning and understanding of what kind of neurons will be needed to recover the memory. We have great pharmacological effects, infusion of antagonists in order to make our brain recover behaviors. But this does not mean a simple effect in the synapses that I am imagining. There are many effects happening, but I think that this is really the beauty of the study of neuroscience. We are studying something that is very complex, that we know a lot of something of those phenomena. And the more we investigate, we make research, we discover, we know that we have even more to discover and to research and investigate.
I'd like to close the session.
Eu gostaria então de
almost time. I give you the word for saying goodbye. I do hope that Izquierdo would be happy with this small tribute. I think that the talks were very stimulating. The pathway he start building is very far from make us happy with all answers, because there is a lot of things that we need to understand about the brain and memory and all the connections. But I'm extremely happy to know that the work he has been doing during his life has stimulated us to think about it and to look forward to the young generation asking, bringing us new answers and so on. I just want to thank you, thank you, Academy. I'd like to know first if Professor Luiz Davidovich would like to say anything, or you would like to say something to each other. Okay, let's start with Beatriz, and then Diana, and then Carlos, and then Luiz Davidovich. I never know, Luis, how to say if the Russian way or the Brazilian way, you know, your surname, but I think that you don't mind quite. Okay, Beatriz, if you just want to say bye-bye to your colleagues and friends.
Thank you. As usual, Brazilian are like Italians. They're warm, welcoming, affectionate people, which I miss very, very much. And I'm really planning to visit Ivan and all my dear friends. And I would I'll be very pleased if you keep me posted on what happens on the Academia Brasileira. because, you know, this is a very interesting, I don't know if it's open to also to people, to lay people, let's say, that don't belong to the academia. Maybe I can just follow on Facebook or whatever. That would be for me important, just to touch base with you all and be ready to go back to Brazil and maybe come to Sao Paulo, which I've never been to reach you. And maybe I can give you a talk on the evolution of the histaminergic system. And of course, it was an immense pleasure and an honor to meet you, to see you again. And I really hope we have another occasion to get together. Thank you. Thank you. Thank you from the depths of my heart.
Thank you. Diana.
Well, I'm grateful and glad for the invitation and the possibility of homage Professor Ivan Izquierdo. We miss him a lot, and actually he was very determinant and relevant in the development of our careers. Many people from Córdoba and many people from Buenos Aires have been working with him and with his disciples, and I continue working with people from in the Urgis, like George Kilfeld, and this is incredible because it's all over the world, the ideas and the concepts he left us, and well, I'm very touched because of this. And well, thank you very much. I enjoy all the talks, and this was a nice journey. And I hope the questions were well answered. I just want to say something about artificial intelligence, connecting with what Carlos Alexandre said, because well, there are simpler activities or functions, like motor functions, that could be replaced by artificial intelligence, and this is a great thing and a great goal. But I don't think that is the same with memory or emotions. It's much more difficult. And well, this is the last thing I wanted to say. Thank you.
Well, I also wish to thank the Academy for the opportunity. I thank to Esper for reaching me out and ask for my participation in this ceremony, in this event. Thank Diana and Beatrice for wonderful talks and for these moments in answering questions. I think we are a nice team. And I think we represent various or different periods of Ivan Izquierdo's career and findings. And for me, it was a lot of emotion also to revisit this theme of memory and novelty and the hippocampus. I am much more on the side of environmental enrichment right now. And so it was good to put my memory to work as well. So thank you very, very much. It was a very nice event. I hope the public has enjoyed as much as I and probably you also enjoyed. Thank you very much.
Professor Luiz, obrigado.
Bom, eu quero agradecer muito a vocês. Eu estou falando em português porque eu sei também que tanto a Beatriz,
that I know that Beatrice and Diana, you do understand Portuguese. As long as I don't get too excited and talk too fast, I promise to control myself, especially in other situations. But I would like to thank you, all of you, for your participation in this event. And most certainly, I did not have the pleasure to work with the Izquierdo, but I was in touch with him for a long time, very dear person, and we miss him a lot in Brazil and in the world with his research, his science, and his knowledge. I would like to thank you all for helping me for the Brazilian Academy of Science to share in sharing with our, well, the general public, members and non-members of the Academy, the science that was developed by Izquierdo, and also in sharing his personality that we knew, that I got to know, and that we miss so much, even with those that did not work directly with him. So once again, thank you so much. Thank you so much, Esper, for organizing this webinar, his moderation, his provocation during this webinar. This is the role of the moderator. I know Esper for a long time. I recall him as president of CNPK and everything he done. And we see this as military mandatory service for science. Thank you so much once again. And I would also call your attention for our next event next Tuesday at 4 o'clock Brazilian time. The theme would be environment, ecologies. And 2 weeks from now, May 25th, we will have another webinar on algorithms, and the title is Provocatives. The provocatives, algorithms lead society, and does the reverse happen also? What will happen later? So good evening. Think please on what was discussed today. Please have a very good weekend. Thank you so much for your attention, and take care. We are now going through the second wave. We have to control this virus. So please use mask, keep distance, social distance, and take a jab. Take the jab. So let's control this epidemics, and we have to win this pandemic. Thank you so much. Good evening, and see you next time. Thank you. And I would like to thank the Academy for facilitating our job. Thank you. Thank you so much. Thank you. Very efficient.